So I'm going to be talking about drug development for Parkinson's disease, and will overview more than a half century of progress and still some of the challenges ahead to make for better treatments for this disorder. Now, the therapeutics of Parkinson's disease has arrived in several waves of drug development and other treatment options over the past half century. And while the main thrust has been symptomatic control of Parkinsonism, the pharmaceutical approaches to intervene with the causes of Parkinson's disease and slowing down disease progression has been ongoing since the very first clinical trial of selegiline more than 30 years ago, together with alpha-tocopherol or vitamin E. So in addition to many pharmaceutical developments, including novel means for drug delivery and different study methodologies for Parkinson's disease therapeutics, there's also been a great understanding of how to investigate this disorder better. Participating in clinical trials has been the option of many patients who've benefited from these options. And many challenges nonetheless remain for how the pharmaceutical industry, together with neuroscientists at the basic level in laboratories, advocacy organizations, foundations, and other interested parties, may help to develop the new therapeutics for the future, still facing our challenge of better therapies. Lessons learned from the development of Parkinson therapies may help us to learn more about how to treat other disorders that currently don't have adequate treatments, like dementia. So in the evolution of Parkinson's disease therapeutics, the arrival of symptomatic therapies is now more than 50 years. Levodopa, amantadine, and another compound called N-propylaporphine were the very first in the 1960s to be effectively changing Parkinson's disease from a poorly controlled disorder to a condition which, in the best of circumstances, has very much improved disabilities. In the late 1980s, research into disease-modifying, potentially curative or neuroprotective therapies first began. In the 1990s, the revolution at that time was neuromodulation, using electrical stimulation of the brain, a therapy that is now termed deep brain stimulation, which is routinely used for symptoms not responsive to medications. And even gene therapy has come into play, as it has in other specialties of medicine. In Parkinson's disease, a few of the studies have been successful to date. Other treatments have been tried over the years, including ways to treat the non-motor features of Parkinson's, such as cognitive impairments, drooling, hallucinations, and some of these have also had gains that we use today routinely. Now, unfortunately, the challenge is still there. We don't have a cure, despite more than 24 clinical trials that have targeted this challenge. And several products that have been developed in the past 3 decades, I've listed them here as monoamine oxidase inhibitors, catechol O-methyl transferase inhibitors. These drugs that work together with levodopa haven't been all that important, and in fact, for the price they cost, may not be all that much of a great value. And several entries into the Parkinson's disease therapeutics have been nothing more than Me Too drugs, that is, absolutely duplicating what's already been out there. So many Parkinson's disease patients are challenged by this situation. Medications are expensive. Some of them don't offer all that much. It's certainly a trial and error marketplace for deciding what can do best, and I'm always surprised that drugs that don't offer that much sometimes get the most promotion in terms of selling to patients. And unfortunately, there's something that I would call therapeutic nihilism, not trying available therapies, pushing medication to the maximum to see what it has to offer and to see if there's more to be gained against the various symptoms that people live with Parkinson's. Back more than 200 years ago, James Parkinson, after whom this disorder is described, is credited with a quote from his monograph called The Essay on the Shaking Palsy, which states that until we're better informed respecting the nature of the disease, the employment of internal medicines is scarcely warrantable. Well, this was a situation back in his time, but fortunately today we have some options that are warrantable because of the development of medications based on animal models or just human experimentation. And this is a listing of the drug therapies that we have available ever since the 1940s, but most importantly, the drug levodopa, which came into clinical use back in 1967, and today still is the mainstay of therapy. improving the effects of levodopa amidst all the other treatment options is still one of the greatest challenges, because the irregularity of its absorption, as you'll see shortly, is still one of the bigger problems in managing Parkinson's as the years go by. So let's take a look at this interesting molecule, which is nothing more than an amino acid celebrating its 50th anniversary of clinical use, and which has a very simple role to play. It is converted in the brain to a neurotransmitter, a substance that signals between nerve cells by an enzyme that is present in the brain and only needs this precursor to go to work and relieve symptoms. The enzyme L-aromatic amino acid decarboxylase, that's quite a mouthful, but that's present in the brain and can actually be augmented by a newly developed gene therapy program that's helping to improve the conversion. But levodopa has a big challenge in getting to the brain. Like many other neutral amino acids or things that are in our normal diet, to get to the brain, it has a number of hurdles to get over, and these are illustrated in this slide, namely that the drug has to be absorbed in the small intestine after having been swallowed and getting to the stomach, getting out of the stomach, being transported once it's absorbed in the GI tract, past the liver, past the lungs, past the blood-brain barrier to get into the brain. And it's no surprise that this drug has a lot of irregularity, as we'll see in a moment, in terms of going to work and helping Parkinson's symptoms. So although it's the most efficacious drug for symptomatic relief of Parkinson's, as the years go by, its effectiveness can become increasingly irregular. And unfortunately for many patients, the delay or failure in onset of levodopa effect from a particular dose is a very common daily experience for many people with Parkinson's. The initial therapy with it can be pretty consistent and quite remarkable in how well it works to almost abolish all symptoms of the disease. But after 3 to 5 years, one or more patterns of motor fluctuations can occur, and sometimes the most troublesome features of living with Parkinson's. So we'll talk in the next few moments about some of the management strategies that are used to improve things, including some therapies that are under development and may become available in the next few years. So I put up a graph here which shows what's happening in your bloodstream when a levodopa tablet is taken. The carbidopa-levodopa combination is the most common product. It's called Sinemet in terms of its brand name formulation, although it's today mostly used as a generic drug. And on the left, the rise and fall of levodopa effect nicely correlates to what many patients experience as the years go by. In other words, a symptom control that is long duration over many hours, and this line illustrates 5 to 6 hours of effect in a patient who is experiencing good response to the levodopa. Unfortunately, after 3 or 5 years, sometimes is replaced by what's termed the short duration response. That is, within 2 to 3 hours, as the blood level of the drug drops off, so does the clinical benefit, as illustrated with that blue line. And one other things that we've learned from research with levodopa is that it seems to have a threshold of effect. The graph here on the left shows improvement in tapping your fingers as a correlate to what Parkinson's symptoms can be. And on the bottom line, you see the increase of blood concentration. So this is showing that there is a dose effect that occurs when you hit a certain threshold, as shown in that red circle. That with a level of the drug in your bloodstream below that effective level, nothing happens. The increase of tapping doesn't occur. The improvement of Parkinson's symptoms doesn't occur. And so there's actually a pretty good correlation between hitting that level, as illustrated in the graph on the right, of improvement in tapping time as a correlate of control of Parkinson's as compared to blood levels. In this case, you see about 180 minutes of improvement in tapping speed related to the blood level. So this patient seemingly was able to store up the effect for 3 hours, even though the blood level dropped off sooner. But perhaps a year later or 2 or more years later, there might be a direct correlation between the blood level and the control of Parkinson's symptoms. So this is what patients often live with this condition. Some of the therapies we have, like the drug entacapone, can extend the duration of action of levodopa. So you see in the blue line an extension of its blood level effect as compared to not taking entacapone in red. And there's the typical threshold illustrated for the effect of the drug. Well, on the right side, you see now a graph of the clinical improvement, the total Parkinson rating scale, and the blue line shows further improvement because of extending the duration of action and the potency of levodopa because of this added drug. So this is the role of adjunctive therapy illustrated with the drug entacapone here. Now, unfortunately, one of the problems of medications for Parkinson's is they're just too irregular to give a consistent effect. In both of these graphs here, you can see 17 patients' blood levels shown. As you're becoming experts in terms of interpreting this, you can see that there's a problem there, that the 2 doses that are given in each of these graphs, as illustrated by that blue arrow pointing downward, show there's a lot of chaos going down. The blood levels rise, but they don't do so all that regularly. On the top left is Sinemet CR, a sustained release product of carbidopa and levodopa. On the lower right is a product called Stalevo, which has entacapone mixed in. And in each instance, among these 17 patients who are illustrated, there's tremendous variability. Some of the doses seem to fall below the threshold line for getting a clinical effect. In other instances, nothing happens, or else you have these huge surges in blood levels, which then translate to the brain and can cause involuntary movements or other side effects. So you can see that even 2 extended duration products on the market today don't do a great job of helping to control symptoms, at least reliably. There is a product out there called Duopa, which has been used for controlling motor fluctuations to keep a more constant blood level, but it involves putting a tube into the stomach, as illustrated here in this picture, with that tube threaded down into the small intestine so that a liquid form of the 2 ingredients of carbidopa, levodopa are continuously delivered to the small intestine. And this can lead to improved control. Now that you can see on the left the variability in sustained release, carbidopa, levodopa, or Sinemet CR, is very much improved in the same patient, and the panel seen on the right where the constant infusion of drug is contrasted to what it was on the oral sustained release product before this therapy was started. So you can improve the consistency of effect, but it comes at a price. You have to have a pump hooked up, and it's a very expensive therapy, not to mention one that's a little bit cumbersome. Other products that are out there include the product called Rytari, which is another sustained release product. And here you can see in these graphs in the black line a much more sustained duration of effect than the immediate release carbidopa levodopa product that is currently the generic standard of care for most patients. So those blood levels look a lot better, at least in this slide from data that was published in 2015. And so you might think, well, this is the end of the problem as long as you take doses of medicine every 5 or 6 hours. And I wish that were the case. But the real data from this indicates that, in fact, there is a lot of variability. When you see those horizontal graphs, they look pretty good, but looking at the vertical lines, that indicates the variability in blood levels from patient to patient or dose to dose. And the reality is that even Rytari, the sustained release formulation, has a lot of potential variability even in switching over from the immediate release product. So we have a big challenge ahead for improving uptake. Well, I'll show you some of the ideas for therapeutics of the future. Improving levodopa by making it a prodrug, linking it to a molecule like sugar to get absorbed better has been one of the studies that's been moderately successful, and we hope that this therapy will continue to be developed. Another strategy is currently under research, which is called the Accordion Pill, which uses a novel way of putting drug into little polymer sheets that are crumpled up into a capsule, accordion pleated fashion, to slowly release throughout the day. And since you're becoming experts in interpreting blood levels, you can see there is a difference between what's illustrated here, the immediate release product taken at 4-hour intervals. You can see the rise and fall and the variability from dose to dose in this sustained release, this immediate release formulation. And let's contrast this to the sustained release accordion pill now taken at 8-hour intervals. You can see a much more constant level illustrated in that purple line. And the implications of this is that we may have a product that will give much more sustained benefit in the future when this product ends up on the market, if it does. In addition, there's studies ongoing currently with a subcutaneously infused form of carbidopa and levodopa put into liquid form. And this study is showing very constant blood levels, which will translate into better control of symptoms. And this kind of formulation is currently under research as well. Perhaps the ultimate product for this continuous infusion won't be a pump, but rather a patch pump that can be put against the skin every day with great ease in terms of delivery of a constant level throughout the day. So here you see another option for treating Parkinson's disease with levodopa. This is a therapy that's using the pulmonary absorption of the drug as a strategy for rapid effect, almost a rescue from being in an off state if somebody has motor fluctuations. And in the red, you can see how quickly inhaled levodopa absorbed through the lungs can occur. The level of therapeutic effect is reached within 5 to 8 minutes, so it seems, and much more rapidly than the oral absorption of the drug is illustrated in the blue lines. Here you can also see how slowly levodopa is absorbed when somebody has a meal in their stomach, in contrast to the light blue lines, which show the rapidity. But typically, it's at least 15 to 20 minutes before a therapeutic level is achieved with the intake of levodopa, in contrast to inhaling it through the lungs. And here you can see an illustration of the speed of onset of effect within 10 minutes, in contrast to placebo. By the way, placebo effect is always present in Parkinson's studies, as illustrated by the black line on top. Patients improve just because the perception that they're doing better because of a new treatment. And this is one of the challenges for research that you can see nicely illustrated here. But certainly inhaled levodopa may turn out to be a very useful therapy for rescue, especially when there's a long delay of oral uptake of the drug. So now let's turn to another form of therapy that's been available for more than 30 years now, drugs that are termed dopaminergic agonists. And those of you on ropinirole or pramipexole can recognize these therapies that are seen amongst others that currently aren't widely used, such as bromocriptine or pergolide. Rotigotine is the dopaminergic agonist that is absorbed through the skin. And it can be said that most of these dopaminergic drugs are very similar in how they work and help somebody with Parkinson's disease. They definitely are effective by themselves as a monotherapy, and generally, though, their best use is combined with levodopa to increase on time. This is the established or approved profile of these drugs. But they also may have unestablished or non-approved uses that can be quite useful, and which over the years have become important for dealing with more advancing disease. For example, to achieve additional anti-tremor effect or to deal with the problem of freezing of gait. Many physicians will give a trial of these medications early on in the course of Parkinsonism, but their greatest role is generally after several years of therapy when levodopa by itself isn't doing the job that's desired. Here you see one of the therapies under development in the United States, the drug apomorphine, which is not a narcotic, even though it has morphine in its name, is a drug that can be infused under the skin continuously over time to achieve more constancy of effect. And in current studies, this small pump hooked up to a small needle under the skin is being shown how it might achieve continuous therapy in a future marketed form. There are other medications currently under development which have some exciting potential. For example, this class of drugs, which are called A2A adenosine receptor antagonists, suggest that we have another way of treating Parkinson's disease other than acting on the dopamine system, the one that levodopa and the dopaminergic agonists act upon. This class of drugs has undergone research for more than 15 years, and this is the only one still remaining in clinical trials. It's currently on the market in Japan, and the promise that it offers is improving the consistency of on time, that is, lessening of off time, times when Parkinson's symptoms emerge. And here you can see contrasted with the placebo effect in left in one of the studies, In blue, on the right side, you can see the effect of improving off time by more than 10% of a typical day's off time for patients having motor fluctuations. So we'll see if this class of drugs or this particular drug comes on the market in the United States, but it offers one more idea of how Parkinson's symptoms can be treated. And finally, we come to more invasive kinds of therapies that are currently under development. And this wiring diagram is too complicated to go through right here, but it does suggest that we have different targets in the brain, different regions of the brain where therapies with medications or with electrical stimulation, in the case of deep brain stimulation, can be helpful. And the adenosine A2A antagonist that was just illustrated has a target where we'll show you right here with this arrow. And there are other therapeutic approaches which go beyond the original circuitry of replacing dopamine, which is what levodopa does. So electrical stimulation of the brain has been widely used throughout the world. We offer it here in this community, and it is a way to treat tremors, involuntary movements, and really all aspects of Parkinson's if medications are doing the thorough job. And here you see a picture of the brain, an MRI scan, with those findings illustrated here, which are the implanted electrodes on the right and left side in a particular target called the subthalamic nucleus. And it's at this location where symptoms of tremor, where slowness of movement, and other aspects of Parkinson's can be controlled beyond the best that medications can offer. This has become a routine therapy. It's always getting better, but for many patients who've been on it, it's been a tremendous improvement over the best that medications can offer. And yet the same target in the brain where those electrodes are placed have been also a target for gene therapy, which is illustrated here, that is linking onto harmless viruses small segments of DNA that can transform the functions of the nerve cells in Parkinson's disease to change their chemistry, to change their functions, so that we can restore function that's been lost as the disease progressed. And here you see an illustration, not to belabor the chemistry here, but you can change the chemistry of the brain. You can use an enzyme, DNA for glutamic acid decarboxylase on this virus transport to change the chemistry of a particular part of the brain so that the signaling between nerve cells is dramatically changed. And the result of that can be improvement in Parkinson's symptoms. In this graph on the upper right corner, as you see here, pointed out on the top is the improvement in Parkinson's scores from the sham operation, which was not really a placebo treatment, but rather just trying to convince the patient that they might be on the therapy. And the lower line, the blue line illustrated here, indicates the level of improvement in Parkinson's scores, a clinically meaningful change that followed this approach to gene therapy. This is not commercially available at this point, but it is an indication that we can get inside the brain and change many of the pathways that were illustrated earlier, rather than just using medications as a way to improve symptom control. And finally, the notion of a neuroprotection against disease progression has been the overwhelming challenge. We'd love to see this disease go away, and there's some very good ideas from the laboratory that have been translated to clinical trials, and these are just a few of them that are currently ongoing and are part of a huge international effort to come up with what might be the causes of the disease, and even if we can't find the cause, at least interrupt the final pathway that causes nerve cells to be lost. So among these are a drug, nilitinib, which is a drug that came from the cancer world and seems to have the potential to slow down the disease. Exenatide is a drug from the diabetes world that also seems to have the potential to improve Parkinson's disease by slowing down loss of nerve cells. Another drug that's been repurposed is isradipine, which is a drug that previously has been used for treating hypertension, but has the potential to slow down the loss of nerve cells. So each of these examples is something that looks promising, has to be studied in clinical trials, and after 2 or 3 years of testing in sometimes hundreds of patients, will ultimately give us a readout in terms of whether these have been effective or not. And all of these studies are currently underway. Even water with enhanced hydrogen content seems to have the potential for improvement, and there's been a preliminary study suggesting this to be effective. Inosine is a current clinical trial drug that has the possibility of improving the disease by increasing concentrations of uric acid in the bloodstream. This study is ongoing right now and is based on a simple observation that people who have higher concentrations of uric acid in their bloodstream seem to have less risk for getting Parkinson's disease and have slower progression if they do have the disorder. So based on something as simple as an observation like that leads to a clinical trial, and we'll learn in a few years whether this has been successful or not. And finally, the approach of getting rid of that bad protein that seems to be so much the cause of the loss of nerve cells, the final pathway, the formation of alpha-synuclein, the misfolded protein that is found in every nerve cell that's degenerating in Parkinson's, itself offers a useful target. And using immunotherapy, that is your immune system targeting these cells with laboratory-generated antibodies, which are called monoclonal antibodies, similar to vaccination. This is an approach that's currently undergoing study around the world as well. And the idea here is that these abnormal proteins that accumulate to form the structures in the brain called Lewy bodies may be something that can be disarmed and ultimately chewed up by the immune cells. Here you see in the lower panel the alpha-synuclein cartoon of misfolded protein, which in the middle panel here shows its accumulation into the Lewy body, which is the structure of the brain that is damaging to nerve cells and cause them to be lost. Well, the idea of a monotherapy with monoclonal antibodies against alpha-synuclein is that we may be able to shut down this process of gradually damaging nerve cells and preserve the population in a patient newly diagnosed. So this study is ongoing and we'll hopefully have some results in the next year or two. And this illustrates the notion that perhaps alpha-synuclein itself is well worth shutting down because it seems to spread from nerve cell to nerve cell, at least in animal models of this disorder. So that perhaps immunizing somebody early in life against alpha-synuclein spread perhaps will be useful, and such a study is ongoing in Europe using standard immunization techniques that are used to prevent infectious diseases. Perhaps this same strategy might be helpful in Parkinson's disease as well. So I think the important thing that we've talked about in treating Parkinson's disease is that we have the challenge ahead of us, a lot of enthusiasm from the scientific community, animal models that help us to translate from what the symptoms are as illustrated in an animal model, even in fruit flies, as indicators of the potential for new therapeutics. And one of these days, we're going to hit it right. But certainly, as you saw earlier, a number of practical applications of new therapies are imminently out there, perhaps as soon as 1 or 2 years from now, and we hope that you'll all be beneficiaries of that.